Imagine for a moment a world so small that it exists beyond the reach of our ordinary sight, a realm not measured in meters or even millimeters, but in micrometers, millionths of a meter. Here, life unfolds in ways both ancient and delicate, invisible to the naked eye yet vital to the story of our planet. This is the world of microbiology, the science devoted to understanding life at its smallest and most fundamental scale. Microbiology, in essence, is the study of organisms too small to be seen without the aid of a microscope. These tiny beings, collectively called microorganisms, or simply microbes, are remarkably diverse. Some are familiar to us in broad terms: the bacteria that live quietly in soil, the yeast that help bread rise, the algae that lend oceans their gentle green shimmer. Others are less familiar: archaea that flourish in the most inhospitable environments, or single-celled protozoa drifting in droplets of pond water. And at the very edge of what we call life, there are viruses, simple yet mysterious entities that require the machinery of living cells to reproduce. These organisms have been present on Earth for billions of years, long before the first animals walked on land, long before plants unfurled their leaves toward the sun. In fact, microbes are among the earliest forms of life we know of, and they have shaped the planet in profound ways. They have helped to create the air we breathe, the soils that sustain forests and fields, and the nutrient cycles that keep ecosystems balanced. Without them, life as we know it could not exist. One of the most humbling truths in microbiology is that microbes are everywhere. They dwell in the deepest trenches of the ocean and in the thin, cold air atop the highest mountains. They cling to grains of sand and swirl in the currents of rivers. They live in clouds drifting far above the earth, and in the warm, sheltered hollows inside our own bodies. Every handful of soil contains more microbes than there are people on Earth. Every drop of seawater teems with life so abundant that it seems almost impossible. And yet, most of these organisms go about their existence without us ever noticing them. It is easy, in the quiet hours of thought, to imagine microbes as a hidden network, an invisible foundation upon which all visible life rests. Some form intricate partnerships with other organisms, offering nutrients or protection in exchange for shelter. Others live independently, carrying out the same essential processes that all life depends upon, capturing energy, building new molecules, and reproducing their kind. They are astonishingly adaptable, able to survive in freezing ice, boiling water, intense radiation, and crushing pressure. In many ways, they are the planet's greatest survivors. Despite their small size, microbes have an outsized influence. They help to recycle the building blocks of life, breaking down fallen leaves in a forest, returning carbon to the atmosphere, and minerals to the soil. In the oceans, photosynthetic microbes contribute a significant portion of the oxygen we breathe. Some live within animals, including ourselves, forming what we call microbiomes, complex communities that play a quiet but essential role in health and well-being. The study of microbiology opens a window into this unseen world. It is a discipline that blends curiosity with careful observation, bringing into focus the delicate forms and graceful movements of tiny creatures. Through the lens of a microscope, a single drop of pond water becomes a landscape, a bustling city of invisible citizens, each carrying out the daily work of living. To watch them is to glimpse life stripped to its essentials. No grand structures, no towering trees or running animals, just the quiet persistence of existence. In this way, microbiology offers more than scientific knowledge. It offers a sense of perspective. It reminds us that life exists on many scales, and that size does not measure importance. The smallest beings are as crucial to Earth's balance as the largest whales or the tallest redwoods. They are, in many respects, the quiet architects of the biosphere, crafting the conditions that make our own lives possible. And so, As we begin our journey into the fundamentals of microbiology, we step gently into a world that has been here since the dawn of time, a world that thrives in silence and invisibility, yet touches every breath we take, every bite of food we eat, every sip of water we drink. In the stillness of this study, we will explore their forms, their behaviors, and the elegant systems they have created to endure. We will move slowly, allowing each idea to unfold like a page turning in an ancient book, letting the quiet rhythms of the microscopic world guide our understanding. This is the beginning, a soft invitation to look closer, to imagine more deeply, and to remember that even the smallest forms of life have stories worth telling. As we move onward, we will meet the inhabitants of this hidden realm in greater detail. But for now, we rest in the knowledge that the world of microbiology is vast, intricate, and endlessly patient, waiting for us to listen. 2. The nature of microorganisms. When we speak of microorganisms, or microbes, we are speaking of life at its most compact scale, life contained within forms so small that a single drop of water can hold millions of them, each going about its own quiet existence. They are not a single group, nor do they all share the same way of life. Rather, the term microorganism is a gentle umbrella, sheltering a remarkable diversity of beings. Some microbes are solitary wanderers, drifting alone through water or air. Others live in vast communities, layered in complex relationships, bound together by mutual needs. Many are single-celled, each cell an entire organism, performing every function needed for survival. Others, like certain fungi and algae, may be made of many cells linked together, forming delicate filaments or small colonies. But whether alone or in groups, their size remains humble. Most range from 1 to a few micrometers, a scale where hundreds could line up across the width of a single grain of sand. We often begin by grouping microorganisms into broad types, though even here, nature resists strict categories. Bacteria are perhaps the most familiar. They are prokaryotic, meaning they lack a nucleus and most of the internal compartments that larger, more complex cells possess. Yet within their tiny boundaries, they carry all the instructions and machinery needed for life. Some live on sunlight, others on chemical energy drawn from minerals, and still others on organic matter left behind by other organisms. They take on simple shapes, spheres, rods, spiral shapes that have persisted for billions of years. Alongside bacteria, there are the archaea. These are also prokaryotic, but they are distinct in their chemistry and evolutionary history. Many live in extreme environments where few others can survive. Boiling springs, hypersaline lakes, acidic pools. Their very existence reminds us that life is capable of enduring conditions that seem utterly inhospitable. In ancient times, they may have thrived in the young Earth's harsher climate, helping to lay the foundation for all later life. Eukaryotic microorganisms represent another step in complexity. Their cells hold their DNA inside a nucleus, and they contain specialized compartments, organelles that perform distinct tasks. Within this group we find protozoa, single-celled organisms that can move gracefully through water using tiny hairs or whip-like tails. Some hunt bacteria or algae, others absorb nutrients directly from their surroundings. We also find microscopic algae, which, like plants, capture sunlight to produce energy. And there are the microscopic fungi, such as yeasts, which quietly ferment sugars into alcohol and carbon dioxide, shaping the making of bread, wine, and countless other foods. And then, on the very edge of what we call life, there are viruses. So small they cannot be seen even with a regular light microscope, viruses exist as tiny packets of genetic material enclosed in protective shells. They cannot grow or reproduce on their own. Instead, they must enter the cells of living organisms and use the host's machinery to make more of themselves. Whether they should be called alive is still a subject of thoughtful debate, but their influence on life, from shaping evolution to moving genes between species, is undeniable. Despite their diversity, all microorganisms share a few fundamental qualities. They require energy, in one form or another, to sustain themselves. They interact with their environment sensing changes, responding to chemical signals, moving toward favorable conditions, or away from harmful ones. They have the ability to grow, to reproduce, and to pass on the instructions for their form and function to the next generation. And they exist in numbers so vast that it is difficult to truly imagine. Microorganisms are found in every corner of the earth. They drift in the upper atmosphere and thrive deep beneath the ocean floor. They inhabit the frozen soils of the Arctic tundra, the boiling waters of volcanic springs, and the lightless depths of underground caverns. They colonize the surfaces of plants, the skins of animals, and the mineral grains of desert sands. Many live within the bodies of larger organisms as peaceful residents, mutual partners, or in some cases as unwelcome guests. The astonishing adaptability of microbes comes, in part, from their simplicity. Small, rapidly reproducing, and able to exchange genetic material in flexible ways, they can adjust to changing conditions far more quickly than larger life forms. What might be a catastrophic shift for an animal or plant, a change in temperature, a new chemical in the environment, a sudden shortage of resources is often an opportunity for microbes to evolve, to explore new strategies for survival. In this sense, microorganisms embody both stability and change. As a group, they have existed for billions of years, shaping the planet's chemistry and supporting the rise of complex life. As individuals and populations, they are in constant motion, dividing, mutating, adapting. They form a living current beneath the surface of the visible world, a quiet and continuous presence that connects every ecosystem. In the study of microbiology, to know their nature is to understand that life is not only what we see, it is also the invisible breath of the planet, the tireless work of countless unseen cells, keeping the great cycles of nature in motion. They remind us that life is both vast and intimate, and that even the smallest beings are woven into the fabric of existence. 3. Microscopic worlds and their discovery. For most of human history, the smallest forms of life existed beyond our imagination. We could feel the warmth of the sun, watch the waves roll in from the sea, and study the flowering of plants. We could observe animals in their movements, from the slow crawl of a snail to the powerful leap of a gazelle. Yet beneath all of this visible life, hidden in plain sight, there was another world. A world no human could see unaided. A world of intricate, delicate structures that shaped the air, the water, and even the soil under our feet. This hidden world is not an invention of modern science. It has always been here for as long as there has been life on Earth. But for countless generations, it lay entirely outside human perception. Without the ability to magnify, our eyes could not witness its existence. For all our curiosity about the world, the microbial realm remained invisible, silent, and unknown. The veil began to lift in the 17th century. In Europe, glassmakers had long been experimenting with lenses, pieces of carefully shaped glass that could bend light in predictable ways. These lenses, first used for spectacles, opened the possibility of exploring very small things in far greater detail than the naked eye could allow. A few curious minds began combining these lenses to create primitive microscopes. What they found was not simply a closer view of familiar things, but an entirely new landscape, hidden within the smallest details. One of the earliest pioneers in this exploration was Robert Hooke, an English scientist and polymath. Hooke's mind roamed widely. He studied physics, mechanics, and astronomy, but he was also deeply fascinated by the structures of living things. In 665, he published Micrographia, a remarkable book filled with detailed illustrations of the world seen through his microscope. Among his observations was the structure of cork, which he described as being made up of tiny, box-like compartments. He called them cells, likening them to the small rooms, or cellula, used by monks in monasteries. Hooke's cells, as we now know, were the empty walls of dead plant tissue, but his naming of them planted a seed in science, the idea that living organisms might be built from repeated, fundamental units. His work was one of the first steps toward a deeper understanding of biology's architecture. At almost the same time in the Netherlands, another figure was embarking on his own journey into the microscopic world. Antony van Leeuwenhoek was not a formally trained scientist. He was a tradesman, skilled in textiles and comfortable with fine, detailed work. Yet he possessed two qualities essential to great discovery: patience and curiosity. Leeuwenhoek taught himself how to grind and polish tiny glass lenses, producing instruments of remarkable clarity for the time. His microscopes were simple. Each was essentially a single, finely crafted lens held in a small frame, yet they could magnify specimens far more sharply than most other microscopes of the day. Through these lenses, Leeuwenhoek began to examine the world around him in extraordinary detail. In drops of pond water, he discovered tiny creatures moving with gentle spirals or quick darting motions. He called them animalcules, meaning little animals. In the film that clung to his teeth, he found countless minute organisms, thriving in that sheltered space. He examined rainwater, peppercorn infusions, and even the tissues of plants and animals. Everywhere he looked, life appeared in miniature, alive and active. These discoveries were more than scientific curiosities. They suggested a profound truth that the world was alive on a scale far smaller than anyone had imagined, and that this unseen life was not rare, but abundant. The invisible spaces between larger forms were not empty voids. They were teeming with motion, energy, and purpose. Over the next 2 centuries, the microscope steadily improved. Lenses became clearer, instruments more stable, and methods for preparing specimens more refined. By the 19th century, scientists could not only see the shapes of cells, but also distinguish some of their inner details. This greater clarity laid the groundwork for cell theory, developed in the 1830s by Matthias Schleiden, a botanist, and Theodor Schwann, a physiologist. Together, they proposed that all living things are made of cells, and that the cell is the basic unit of life. Later, the work of Rudolf Virchow added a third principle, that all cells arise from pre-existing cells. Cell theory transformed our understanding of biology. It revealed that the same basic building blocks form plants, animals, and microorganisms alike. The living world, in all its variety, was united by a common structural foundation. As the 20th century dawned, another leap forward arrived: the electron microscope. Unlike light microscopes, which rely on visible light to reveal detail, electron microscopes use beams of electrons, allowing scientists to see structures far smaller than the wavelength of light itself. With this new tool, the inner landscapes of cells came into view: delicate membranes, coiled strands of DNA, and the tiny energy-producing mitochondria. The architecture of life, once hidden in shadows, was revealed with stunning precision. In time, other forms of microscopy emerged. Fluorescence microscopy allowed specific molecules to glow with color, tracing their movements through cells. Confocal microscopy provided crisp, three-dimensional images. Cryo-electron microscopy froze delicate structures in place, preserving their shape for detailed study. Each new method added a new layer of depth to our view of the microbial world. Yet even in the earliest days, before these advanced tools, there was wonder in simply seeing. Imagine peering through one of Leeuwenhoek's simple lenses. holding a small droplet of pond water on a thin glass slide. At first, perhaps, you see nothing unusual. Then, with a slight adjustment of focus, shapes emerge, tiny specks moving deliberately through the liquid. Some glide smoothly, propelled by beating hair-like cilia. Others spin or dart forward, driven by slender whip-like flagella. A few drift lazily, carried by the gentle currents within the drop. This is a universe in miniature, a place where life is both ancient and immediate. Each organism, no matter how small, is carrying out the same essential tasks as all living things, feeding, growing, dividing, and passing on its instructions for life to the next generation. The microscope, in this way, became a bridge between worlds. It connected the visible with the invisible, the known with the undiscovered. It allowed us to witness not only the forms of microorganisms, but also their behaviors, how they move, how they interact, and how they adapt. It opened the door to microbiology as a discipline, a path of study that would reveal the deep connections between the smallest beings and the great cycles of Earth's life. And perhaps most importantly, it offered perspective. It reminded us that life is not only what we can see. There is an entire realm of existence in which complexity thrives at scales smaller than our senses can reach. This realm sustains the planet's balance and has done so for billions of years. Without it, there would be no forests, no oceans as we know them, no air fit to breathe. From the early lenses of Hooke and Leeuwenhoek to the intricate instruments of today, the journey into the microscopic world has been one of patient curiosity. Each improvement in our ability to see has brought new questions and new appreciation for the subtle architecture of life. And with every glimpse, we are reminded of the same gentle truth that the smallest lives are no less wondrous than the largest. As we move forward, our next step will be to meet the earliest and most enduring forms of cellular life, the prokaryotes, whose simple structures conceal a remarkable capacity for adaptation and survival. But before we begin, we can pause to remember that the story of microbiology is in many ways the story of seeing, of learning to look beyond what is immediately visible, of discovering that the world is larger and smaller than we ever knew. 4. Prokaryotic cells. Among the inhabitants of the microscopic world, the prokaryotes are the most ancient and enduring. They appeared billions of years ago, long before the first plants rooted themselves into soil or the first animals ventured across the surface of the earth. They were here before forests, before coral reefs, even before the oxygen-rich air we breathe today. Their form is simple in appearance, yet profound in capability, and they remain among the most adaptable and influential life forms on the planet. The word prokaryote comes from Greek, meaning before nucleus, a reflection of their defining feature, the absence of a membrane-bound nucleus. Instead, their genetic material exists as a single loop of DNA, compacted within a central region of the cell, called the nucleoid. There are no internal compartments like the ones found in the more complex eukaryotic cells of plants, animals, fungi, and protists. Yet this simplicity is not a limitation but a refinement. Every part of the prokaryotic cell is devoted to the essential processes of life, carrying them out with remarkable efficiency. Prokaryotic life is divided into two great domains, bacteria and archaea. At first glance, under a microscope, they appear similar. Tiny cells, only a few micrometers long, without visible compartments or internal structures. Yet their histories and chemistries are distinct. Bacteria are perhaps the more familiar, found in nearly every environment on Earth, from the soil beneath our feet to the water we drink, from the air we breathe to the surfaces of our own skin. Many are free-living, drifting in water or resting on solid surfaces, quietly conducting their lives. Others have formed intricate relationships with larger organisms, some beneficial, others less so. Archaea, on the other hand, are more often associated with extreme environments. They thrive in boiling hot springs, in highly saline lakes, in acidic pools, and in oxygen-free sediments deep beneath the seafloor. Some produce methane gas as a natural byproduct of their metabolism, influencing the chemistry of the planet in ways that extend far beyond their hidden habitats. Though they resemble bacteria in size and shape, their molecular structures are unlike any other form of life. In form, prokaryotic cells are most often spheres, rods, or spirals. These simple shapes, known respectively as cocci, bacilli, and spirilla, are not just matters of appearance but influence how the cells move, attach to surfaces, and exchange nutrients with their surroundings. Every cell is bounded by a delicate yet protective cell membrane, a layer of lipids and proteins that carefully regulates what enters and leaves. Most bacteria also have a rigid cell wall that provides shape and prevents the cell from bursting in fluctuating environments. Some are encased in a capsule, a soft outer layer made of sugars or proteins, which can help them cling to surfaces, resist drying, or evade the attention of other organisms. Many can move, propelled by whip-like flagella that rotate like tiny propellers, allowing them to swim toward favorable conditions or away from harmful ones. Others carry fine hair-like pili, which help them attach to surfaces or even exchange genetic material with neighboring cells. Inside, the cytoplasm contains everything needed to sustain life. The DNA, though not enclosed in a nucleus, is carefully organized and accompanied by smaller loops of genetic material called plasmids. These plasmids often carry special instructions that can give the cell an advantage in certain environments. Scattered throughout the cytoplasm are ribosomes, the molecular machines that read the genetic code and assemble proteins. These proteins serve countless functions, from building cell structures to driving chemical reactions. Even without internal compartments, prokaryotes manage the complex chemistry of life with precision. In many cases, their cell membranes host the enzymes and molecules needed for capturing energy, whether from sunlight, from inorganic compounds, or from organic matter. The diversity of their energy-gathering strategies is one of the keys to their success. Some prokaryotes are photosynthetic, turning sunlight into chemical energy, as cyanobacteria do when they release oxygen into the atmosphere. Others draw energy from inorganic substances such as hydrogen sulfide or ammonia, making them independent of both sunlight and organic food sources. Many obtain their energy by breaking down organic matter, recycling the remains of once living things back into the cycles of nature. Because of this metabolic flexibility, prokaryotes inhabit niches that few others can. They live in deep-sea vents where no light penetrates, in frozen soils where temperatures stay far below freezing, and in the digestive tracts of animals where they help break down food into usable nutrients. Prokaryotes reproduce simply and efficiently through a process called binary fission. The cell grows, copies its DNA, and then divides into two genetically identical daughter cells. Under the right conditions, this cycle can happen astonishingly quickly, allowing populations to expand in a matter of hours. But their adaptability is not limited to reproduction. Prokaryotes are able to exchange genetic information directly, even between unrelated cells, in a process known as horizontal gene transfer. This can happen through the exchange of plasmids via pili, by absorbing free DNA from the environment, or through the actions of viruses that carry genetic material from one cell to another. Such exchanges can give rise to entirely new abilities almost overnight, allowing them to survive challenges that would otherwise be deadly. Their influence on the planet is vast and essential. They are the foundation of nutrient cycles, breaking down dead matter and releasing elements such as carbon and nitrogen back into forms that plants, fungi, and animals can use. In the soil, certain bacteria form partnerships with plant roots, capturing nitrogen from the air and turning it into a form the plants can absorb. In the oceans, cyanobacteria produce oxygen and form the base of food chains that sustain countless other organisms. In the history of life, they have been nothing less than world builders. Billions of years ago, photosynthetic prokaryotes began releasing oxygen into the air, slowly transforming the atmosphere and making possible the evolution of complex oxygen-breathing life. Some prokaryotes have taken survival to an extreme. When faced with harsh conditions, certain species can form endospores, tough, dormant structures that can endure extreme heat, deep cold, intense radiation, and complete dryness for years, even centuries. In this state, they wait patiently until conditions improve, then awaken and resume their quiet work. Others rely on sheer adaptability, changing their metabolism or altering their genetic makeup to match their environment. It is this combination of patience and adaptability that has allowed them to persist through every great change the Earth has known. The story of prokaryotes is a reminder that life does not need complexity to be powerful. These cells are small, lacking the elaborate architecture of eukaryotes, yet they are central to the functioning of the planet. They are living threads woven into every ecosystem, linking the deep past with the present day. They have survived mass extinctions, climate shifts, and geological upheavals, and they will likely endure long after many other forms of life have disappeared. As we continue into the next chapter of our journey, we will meet their more complex descendants, the eukaryotic microorganisms, and see how life evolved from these ancient, simple beginnings into cells with greater internal complexity and new strategies for survival. 5. Eukaryotic microbes. From the ancient simplicity of prokaryotes, life eventually gave rise to a new kind of cell, the eukaryote. These cells, more complex in their internal structure, marked a turning point in the history of life. Where prokaryotes carry their genetic material in a single, unprotected loop of DNA, eukaryotes safeguard theirs within a membrane-bound nucleus. This change, along with the development of specialized compartments known as organelles, allowed for new possibilities in the way life could grow, move, and interact with its surroundings. Though eukaryotic cells include the building blocks of plants, animals, and fungi, there is also a fascinating array of single-celled eukaryotes that inhabit the microscopic world. These eukaryotic microbes are as varied in form and function as their prokaryotic ancestors, and they offer a glimpse into the evolutionary innovations that pave the way for complex life. The most striking feature of a eukaryotic microbe is its nucleus. Here, strands of DNA are organized into chromosomes, each carrying the instructions for building and maintaining the organism. The nuclear envelope, a double layer of membrane, protects this genetic material, allowing for greater control over how genes are read and expressed. This separation of genetic control from the rest of the cell's processes is one of the defining characteristics of eukaryotic life, and it provides a level of regulation that supports more intricate and adaptable systems. Surrounding the nucleus, the cytoplasm contains an array of organelles, each performing specific tasks. Mitochondria, the cell's powerhouses, convert nutrients into usable energy. In photosynthetic eukaryotic microbes, chloroplasts capture sunlight and turn it into chemical energy, much as plants do. The endoplasmic reticulum and Golgi apparatus manage the production, folding, and delivery of proteins, while lysosomes break down and recycle unwanted materials. This division of labor within the cell means that eukaryotic microbes can carry out more complex behaviors than their prokaryotic relatives. Many are motile, moving gracefully through their environment with whip-like flagella or a fringe of tiny cilia. Some extend and retract portions of their cytoplasm, forming temporary projections called pseudopodia that allow them to flow forward in an amoeboid motion. These forms of movement give them flexibility in how they seek out food, avoid danger, or interact with other cells. Within the group of eukaryotic microbes, protozoa are perhaps the most well-known. They are single-celled organisms that live in water, soil, and within other organisms. Some feed by engulfing bacteria or smaller particles, wrapping themselves around the food and drawing it inside. Others graze on algae, while still others live as parasites within animal hosts. Despite their small size, protozoa display a remarkable variety of lifestyles. The amoebas, for example, are shapeless and ever-changing, extending pseudopodia in any direction as they move. Ciliates, such as the well-studied paramecium, are covered in tiny cilia that beat in coordinated waves, propelling the organism smoothly through the water. Flagellates, on the other hand, swim by rotating one or more long whip-like flagella. Each of these strategies has evolved to suit different environments and ways of obtaining food. Another important group of eukaryotic microbes is the microscopic algae. Like the larger plants they resemble, they contain chloroplasts that allow them to photosynthesize. This means they can capture sunlight and use it to build sugars from carbon dioxide and water. Microscopic algae come in many forms, from simple, single cells to elaborate colonies. Diatoms, with their intricately patterned silica shells, drift in the oceans and lakes, forming a major component of the world's phytoplankton. Dinoflagellates, many of which can move with the aid of flagella, are also abundant in marine environments and are key players in the cycling of nutrients. These photosynthetic microbes are more than just passive drifters. They are primary producers, forming the foundation of aquatic food webs and contributing significantly to the oxygen content of Earth's atmosphere. Microscopic fungi, such as yeasts, are also part of the eukaryotic microbial world. While most fungi are multicellular, forming networks of thread-like hyphae, some remain single-celled for most of their life cycle. Yeasts have long been companions to human activity, playing essential roles in the making of bread, beer, and wine. In nature, they help decompose organic matter, breaking down complex molecules into simpler ones that other organisms can use. This process of decomposition is a vital part of nutrient cycling, ensuring that the building blocks of life are continually recycled through ecosystems. Eukaryotic microbes exhibit an extraordinary range of feeding strategies. Some, like the algae, make their own food through photosynthesis. Others are heterotrophs, consuming organic material directly. Still others can switch between modes depending on their environment, a flexibility that allows them to thrive under changing conditions. This adaptability extends to reproduction as well. Many reproduce asexually, simply dividing to create genetically identical offspring. Others engage in sexual reproduction, combining genetic material from two parents to create variation in the next generation. In some cases, they alternate between sexual and asexual phases in response to environmental cues. The origins of eukaryotic cells themselves are a story of cooperation at the deepest level. The widely accepted endosymbiotic theory suggests that mitochondria and chloroplasts began as free-living prokaryotes that entered into a partnership with a host cell. Over time, these internal partners became permanent residents, passing from generation to generation as part of the larger cell. The benefits of such partnerships were profound. The host cell gained powerful new ways to harvest energy, while the symbionts received shelter and a steady supply of nutrients. This merging of capabilities may have been one of the key steps that enabled life to grow in complexity. In their ecological roles, eukaryotic microbes are both shapers and sustainers of the environments they inhabit. In aquatic systems, photosynthetic algae are at the base of food chains, supporting everything from tiny crustaceans to great whales. Protozoa help regulate bacterial populations, preventing them from growing unchecked. Microscopic fungi decompose dead matter, freeing nutrients for plants and other organisms. Many eukaryotic microbes also live in symbiosis with larger hosts, providing benefits such as nutrient exchange, digestion assistance, or even protection against pathogens. While their contributions are often overlooked, eukaryotic microbes are woven deeply into the fabric of life. They connect the microscopic world to the visible one, linking bacterial communities to the plants and animals that depend on them. They remind us that complexity can exist on any scale, and that even a single-celled organism can display intricate behaviors sophisticated structures, and ecological importance. Though invisible without the aid of a microscope, these organisms fill the waters, soils, and even the air we breathe. They drift in ocean currents, cling to the surfaces of leaves, inhabit the gut of a grazing animal, and enrich the soil beneath a forest floor. They are part of the same grand story of life that began with their prokaryotic ancestors, but they have added new chapters of innovation, adaptability, and cooperation. As we continue our journey through the fundamentals of microbiology, we leave behind the single cells of eukaryotic microbes and step towards something stranger, still, the tiny, enigmatic entities that dwell on the very edge of life. In the next part, we will explore viruses, their simple yet powerful nature, and their role in the grand tapestry of biology. 6. Viruses on the edge of life. In the vast and intricate web of life, there exists a category of entities so unusual that scientists still debate whether they can truly be called alive. These are the virus's minute packages of genetic material wrapped in a protective coat, simple in structure, yet capable of exerting profound effects on the living world. Unlike the cellular organisms we have explored so far, viruses do not possess the machinery needed to carry out life's processes on their own. They cannot grow, cannot generate their own energy, and cannot reproduce without entering the living cells of another organism. And yet, despite these limitations, they have woven themselves into the evolutionary history of life, influencing the shape and diversity of countless species over billions of years. A virus is, at its core, a piece of genetic information, either DNA or RNA, encased in a shell of protein called a capsid. Some viruses have an additional outer envelope made from lipids, often stolen from the host cell's own membrane during the process of viral release. The shapes of viruses range from simple icosahedrons, with their 20 triangular faces, to elongated rods, to complex structures that resemble tiny lunar landers. Despite their diversity in form, all viruses share the same fundamental strategy. They must find a suitable host cell, enter it, and use the host's machinery to make copies of themselves. Viruses are far smaller than even the smallest bacteria. Most measure only tens to hundreds of nanometers across, so small that they cannot be seen with a light microscope. It was not until the development of the electron microscope in the 20th century that their forms were revealed in detail. Before that, their existence could only be inferred from the effects they caused and the way these effects could pass through filters fine enough to trap bacteria. The discovery of viruses began in the late 1800s, when scientists studying diseases in plants and animals found that some infections were caused not by bacteria, but by something smaller and simpler. The tobacco mosaic virus, one of the first to be identified, could pass through a filter designed to remove bacteria, yet still cause disease in plants. This observation marked the first recognition of an entirely new category of infectious agent. To reproduce, a virus must first locate and attach to a host cell. It does this by recognizing specific molecules on the cell's surface, much like a key fitting into a lock. This specificity means that most viruses can infect only certain types of cells, and often only certain species. Once attached, the virus delivers its genetic material into the cell, either by injecting it through the cell membrane or by entering the cell whole and releasing it inside. Once inside, the viral genome takes over the host's molecular machinery, redirecting it from its normal duties to the singular task of producing new viral components. The process of viral replication follows different patterns depending on the type of virus, but the outcome is always the same: the assembly of many new virus particles. In some cases, the new viruses burst out of the host cell, destroying it in the process. In others, they bud slowly from the cell's surface, allowing the host to survive for a time while still producing more viruses. Some viruses can insert their genetic material directly into the host cell's DNA, lying dormant for months or years before becoming active again. This hidden phase allows them to persist in a population long after the initial infection has passed. The question of whether viruses are truly alive remains an open one. They possess genetic material, can evolve through mutation and natural selection, and have adapted to survive in a vast range of environments. Yet without a host cell, they are inert, unable to carry out the basic processes of metabolism, growth, or independent reproduction. Some scientists describe them as existing in a gray area between the living and the non-living, calling them replicators, rather than organisms in the traditional sense. Whatever we choose to call them, their impact on the living world is undeniable. Viruses are not only agents of disease, they are also drivers of evolution. By inserting their genetic material into the genomes of their hosts, they can introduce new genes or alter existing ones. Over long spans of time, these changes can influence the traits and adaptations of entire species. In fact, a significant portion of the DNA in many organisms, including humans, appears to have originated from ancient viral insertions. These remnants, now inactive, are silent reminders of past encounters with the viral world. The variety of viruses is staggering. They infect all forms of life, animals, plants, fungi, bacteria, and even other viruses in rare cases. Some cause devastating diseases, while others exist harmlessly within their hosts or even provide benefits. In certain marine environments, viruses help regulate the populations of bacteria and algae, preventing any one group from growing too dominant. This balancing act plays a role in nutrient cycling and the stability of aquatic ecosystems. Despite their association with illness, viruses have also been harnessed for beneficial purposes. In medicine, modified viruses are used to deliver genes into cells in a technique known as gene therapy. By replacing faulty genes or introducing new ones, researchers hope to treat or even cure genetic disorders. Viruses are also employed in the development of vaccines, serving as harmless carriers that train the immune system to recognize and defend against more dangerous versions. In agriculture, certain viruses are being explored as tools to control crop pests, reducing the need for chemical pesticides. From an evolutionary perspective, viruses may be as ancient as life itself. Some theories suggest they originated from fragments of genetic material that escaped from early cells, while others propose they may have been fully independent life forms that gradually lost the ability to live on their own. Whatever their origin, they have coexisted with cellular life for billions of years, adapting alongside it, shaping its diversity, and sometimes guiding its evolutionary paths. In the quiet language of microbiology, viruses remind us that life, or near life, takes many forms. They are neither wholly independent nor entirely inert, existing in a state that challenges the boundaries of our definitions. Their simplicity belies their influence, for they have touched every branch of the tree of life, from the smallest bacteria to the largest mammals. As our understanding of viruses grows, they are part of the invisible currents that flow through the biosphere, subtle yet powerful forces that have helped shape the story of life on Earth. And while much remains to be learned about them, they stand as a reminder that even at the very edge of what we call life, there is still beauty, mystery, and a deep connection to the living world. From here, our journey in microbiology will turn once more to the realm of independent life, exploring how microorganisms gather energy from their environments. We will see how metabolism in all its varied forms powers the hidden engines of the microbial world and sustains life at every scale. 7. Microbial metabolism and energy. At the heart of every living cell, whether it belongs to a towering tree, a swift bird, or the smallest microbe, lies the same essential truth: life requires energy. Energy is the quiet current that runs through every process of existence, from the replication of DNA to the movement of a single cilium, pushing a microbe gently through the water. Without a constant supply of usable energy, life cannot be sustained. Microorganisms, despite their small size, are masters at capturing and transforming energy from their surroundings, and in doing so, they drive the cycles that sustain ecosystems across the planet. The term metabolism describes the sum of all chemical reactions within a cell. It is not a single process, but a collection of interconnected pathways, each carefully regulated to balance the needs of the organism. Broadly, these reactions fall into two categories: those that build and those that break down. Anabolic reactions use energy to construct complex molecules from simpler ones, as when proteins are assembled from amino acids. Catabolic reactions release energy by breaking down larger molecules, such as the digestion of sugars into carbon dioxide and water. Together, these two sides of metabolism form a complete cycle of energy use and renewal, allowing the cell to maintain itself, grow, and reproduce. Microbes obtain their energy in a variety of ways, reflecting billions of years of adaptation to every conceivable environment. Some are phototrophs, capturing the energy of sunlight, much as plants do. Within this group, oxygenic phototrophs, like cyanobacteria and many algae, use light to drive the production of sugars from carbon dioxide and water, releasing oxygen as a byproduct. This process, known as photosynthesis, has been one of the most transformative events in Earth's history, gradually enriching the atmosphere with oxygen and paving the way for the evolution of aerobic life. Other phototrophs are anoxygenic. They still use light for energy, but rely on different electron donors, such as hydrogen sulfide, and do not produce oxygen. These microbes are often found in specialized environments like sulfur-rich hot springs or stagnant water layers where oxygen is absent. Not all microbes rely on light. Many are chemotrophs, drawing their energy from chemical compounds. Some are chemoautotrophs, able to build their own organic molecules using energy from the oxidation of inorganic substances. This group includes nitrifying bacteria that convert ammonia to nitrate in soils, and sulfur-oxidizing bacteria that thrive in the plumes of deep-sea hydrothermal vents. These environments, where sunlight never penetrates, support entire ecosystems based on chemical energy, a testament to the adaptability of microbial metabolism. Other chemotrophs are chemoheterotrophs, which, like animals, obtain both their energy and carbon from consuming organic matter. Many bacteria, fungi, and protozoa fall into this category, playing key roles in decomposition and nutrient recycling. The versatility of microbial metabolism allows life to persist in environments that seem inhospitable. In the cold, dark depths of the ocean, chemotrophic microbes oxidize hydrogen and methane that seep from the seafloor. In arid deserts, phototrophic bacteria cling to the undersides of translucent rocks, using faint light to fuel their growth. Even in the boiling waters of hot springs or the acidic runoff from mines, specialized microbes have evolved pathways to capture and use energy in conditions that would destroy most other forms of life. A central process in microbial energy management is cellular respiration, the controlled release of energy from organic molecules. In aerobic respiration, oxygen serves as the final electron acceptor in a chain of reactions that generate molecules of ATP, the cell's main energy currency. This process is highly efficient, yielding much more ATP than anaerobic alternatives. Yet many microbes thrive without oxygen, using other molecules such as nitrate, sulfate, or even carbon dioxide in place of oxygen to accept electrons. These forms of anaerobic respiration are less efficient, but allow survival in oxygen-poor or oxygen-free environments. Some microbes rely on fermentation, breaking down organic molecules without the use of an electron transport chain at all. Fermentation yields far less energy, but is simple and effective, sustaining life in niches where other pathways are not possible. ATP, or adenosine triphosphate, is central to all these processes. This small molecule stores energy in the bonds between its phosphate groups, releasing it when those bonds are broken. In a microbe, ATP fuels everything from the movement of flagella to the synthesis of cell walls and the repair of damaged proteins. The constant turnover of ATP production, use, and replenishment is a measure of life's ongoing activity. Even the smallest bacterium cycles through countless ATP molecules in the span of a few seconds, a reminder of the ceaseless motion within each living cell. Microbes not only capture energy for themselves, but also influence how energy flows through ecosystems. In aquatic environments, photosynthetic microbes form the foundation of food webs, capturing sunlight and converting it into organic matter that sustains countless other organisms. In soils, decomposers break down plant litter, releasing nutrients that plants and fungi can reuse. In the guts of animals, microbial communities help extract energy from food, breaking down complex carbohydrates into simpler molecules the host can absorb. Without these contributions, the cycles of energy and matter that sustain life would soon falter. One of the most remarkable aspects of microbial metabolism is its flexibility. Many microbes can switch between energy sources, depending on availability. A single bacterium might photosynthesize when light is abundant, then switch to consuming organic matter when in darkness. Others can use different electron acceptors for respiration depending on what is present in their environment. This metabolic versatility is a survival strategy, allowing microbes to endure in places where conditions change rapidly or unpredictably. Energy flow in the microbial world is not isolated from the rest of life. The oxygen we breathe, the carbon dioxide exchanged by plants, the methane released from wetlands all bear the fingerprints of microbial activity. On a global scale, microbes regulate the balance of gases in the atmosphere, influence climate patterns, and shape the chemistry of oceans and soils. They are the unseen engineers of the biosphere, orchestrating the invisible transformations that make Earth habitable. To understand microbes is to see energy not as a single spark, but as a continuous cycle. It begins with capture from sunlight, from chemical gradients, from the breakdown of organic matter, and flows through countless transformations before being dissipated as heat. In the quiet workings of microbial metabolism, this cycle plays out billions of times each second in every corner of the planet. And though each microbe is small, together they form a vast, interconnected web of energy exchange that sustains the diversity of life. As we move forward in our journey through microbiology, we will turn from the general processes of metabolism to the specifics of how microbes grow and multiply. In the next part, we will explore microbial growth and reproduction, the patterns and strategies that allow them to persist, spread, and adapt to the endless variety of environments they inhabit. 8. Microbial growth and reproduction. In the quiet and hidden world of microorganisms, growth and reproduction are the steady rhythms that sustain their presence across the planet. For microbes, these processes are not merely events, but an ongoing flow of life, repeated countless times in every droplet of water, every grain of soil, every breath of air. Growth, in the microbial sense, does not mean an individual cell expanding indefinitely, but rather an increase in the number of cells within a population. Reproduction ensures the continuity of these populations, allowing microbes to persist, adapt, and spread into new environments. For most bacteria and archaea, reproduction is accomplished through a process called binary fission. It is a simple, elegant cycle. The cell first grows, increasing its internal components and duplicating its DNA. The genetic material, arranged in a circular chromosome, is carefully copied so that each new cell will receive an identical set of instructions. Once replication is complete, the cell elongates, and a partition forms in the middle. Gradually, This partition, known as a septum, closes, dividing the cell into two separate entities. Each is a complete, fully functional cell, ready to live independently. Under ideal conditions, this process can be remarkably fast, with some bacteria capable of doubling their numbers in as little as 20 minutes. While binary fission is the primary method of reproduction for many prokaryotes, variations exist. Some microbes reproduce by budding, in which a small new cell forms as a protrusion from the parent before detaching. Others produce chains or clusters of cells, remaining connected after division. Certain cyanobacteria form filaments that grow by repeated cell division, sometimes specializing different cells for specific roles, such as nitrogen fixation. This diversity of growth patterns reflects the adaptability of microbial life to different ecological niches. In eukaryotic microbes, reproduction can be more complex. Many single-celled algae and protozoa also divide by binary fission, but others employ mitosis, a process in which the cell's chromosomes are carefully organized and separated before division. Mitosis ensures that each daughter cell receives an exact copy of the genetic material, maintaining the stability of the species over time. Some eukaryotic microbes reproduce by multiple fission, in which the nucleus divides several times before the cell splits into many offspring at once. This method can produce bursts of new individuals, quickly increasing population size when conditions are favorable. Microbial reproduction is often closely tied to environmental conditions. When nutrients are plentiful, populations can grow rapidly, following what is known as an exponential growth pattern. Each cell division doubles the number of cells, leading to a steep rise in population size. However, this rapid growth cannot continue indefinitely. As resources are consumed and waste products accumulate, growth slows and eventually reaches a plateau. This balance between growth and limitation is reflected in the microbial growth curve, which moves through distinct phases: lag, exponential, stationary, and death. In the lag phase, newly introduced cells adjust to their surroundings, preparing the internal machinery needed for division. In the exponential phase, conditions are ideal, and cells divide at their maximum rate. The stationary phase marks the point where growth and death occur at roughly equal rates, as resources become scarce. Eventually, in the death phase, conditions deteriorate to the point where more cells die than are produced. Some microbes have evolved remarkable strategies to survive periods when growth is no longer possible. Certain bacteria form endospores, tough, dormant structures that encase the cell's genetic material and essential components. In this protected state, the organism can endure extreme heat, freezing, drying, or chemical damage. When favorable conditions return, the endospore germinates, releasing a fully functional cell ready to begin dividing again. Other microbes enter less extreme dormant states, slowing their metabolism to a bare minimum until resources improve. These survival strategies allow microbial lineages to persist through harsh and unpredictable environments. Reproduction in microbes is not solely about producing identical offspring. Genetic variation, though often subtle, is crucial for long-term survival. While changes in the DNA sequence arise naturally during replication, many microbes have additional ways to exchange genetic material. Through processes such as conjugation, transformation, and transduction, they can share genes with unrelated individuals, even across species boundaries. This horizontal gene transfer introduces new traits into populations, sometimes granting advantages like resistance to environmental stresses or new metabolic abilities. Though not reproduction in the strict sense, these exchanges are deeply intertwined with microbial growth and adaptation. In some eukaryotic microbes, sexual reproduction plays an important role in generating diversity. This process involves the fusion of genetic material from two different cells, creating offspring with combinations of traits not found in either parent. In algae, for example, sexual reproduction may occur when environmental conditions become challenging, producing resistant spores that can survive until conditions improve. Protozoa, like the ciliate paramecium, also engage in forms of sexual exchange, swapping genetic material before resuming normal cell division. Though sexual reproduction is generally more complex and energy-intensive than asexual methods, it offers the benefit of greater genetic flexibility. The speed and efficiency of microbial reproduction have profound implications for ecosystems. In aquatic environments, photosynthetic microbes can rapidly multiply when sunlight and nutrients are abundant, forming blooms that feed entire food webs. In soils, bacterial populations can surge after rainfall, accelerating the decomposition of organic matter and releasing nutrients for plants. Even within the human body, microbial communities can shift quickly in response to changes in diet, health, or environment. This rapid turnover allows microbial populations to respond to ecological changes far more swiftly than larger organisms can. Yet growth is always balanced by limits. Microbes are bound by the same physical and chemical constraints as all life. They require energy, water, and suitable chemical conditions to thrive. Competition with other organisms, predation by protozoa or viruses, and changes in temperature or pH can all slow or halt growth. The interplay between growth-promoting conditions and limiting factors shapes the size, diversity, and function of microbial communities. When we consider microbial reproduction, it is easy to think only of numbers, how quickly a population can grow, how vast it can become. But reproduction also represents continuity. Each division, each new cell, is a link in an unbroken chain stretching back to the origins of life on Earth. The cells dividing in a drop of water today are part of a lineage that has persisted through billions of years, weathering ice ages, mass extinctions, and the slow transformations of continents and seas. In every division, the story of life is retold in miniature, passed from one cell to the next. The beauty of microbial growth and reproduction lies in its simplicity and persistence. It is a process that needs no elaborate structures, no complex organs, yet it is as vital to the balance of life as the turning of the seasons or the flow of rivers. In the stillness of a pond or the darkness of the soil, unseen to the human eye, these cycles unfold again and again, sustaining the smallest yet most abundant forms of life. In the next part of our journey, we will turn to the intricate relationships between microbes and other living beings. We will explore the many ways microorganisms interact with their hosts and environments, from the mutual partnerships that benefit all involved to the subtle and sometimes harmful influences that shape the living world. This realm of microbial relationships, the study of symbiosis and the microbiome, reveals how deeply interconnected life is at every scale. 9. The microbiome and symbiosis. In the tapestry of life, no organism exists entirely alone. Every plant, every animal, every human being is part of a network of relationships that bind living things together. At the microscopic level, these relationships are even more intricate, for microorganisms have been forming close associations with other organisms since the earliest days of life on Earth. Some of these associations are fleeting encounters, while others are so deep and long-standing that neither partner could survive in quite the same way without the other. The term symbiosis describes this living together, a broad word that embraces mutual benefit, quiet neutrality, and even gentle conflict. Within this realm lies the concept of the microbiome, the community of microorganisms that lives in and on a larger host, shaping its health, development, and interactions with the world. The microbiome is not a single species, but a collection of many. In a healthy human, for example, thousands of different bacterial species, along with fungi, archaea, and viruses, inhabit the skin, mouth, gut, and other surfaces. Together they form a complex ecosystem, each species occupying its own niche, performing specific functions, and interacting with others in ways that maintain balance. In the gut, microbes help break down complex carbohydrates into simpler molecules that the body can absorb. They produce vitamins, such as vitamin K and certain B vitamins, that we cannot make ourselves. They train the immune system, teaching it to distinguish between harmless companions and harmful invaders. This intimate partnership is the result of a long coevolutionary journey in which both host and microbe have adapted to life together. Symbiotic relationships are not unique to humans. In the plant world, the roots of many species are home to mycorrhizal fungi, which extend fine filaments into the surrounding soil. These fungal threads greatly increase the plant's ability to absorb water and nutrients, particularly phosphorus. In return, the plant supplies the fungus with sugars produced during photosynthesis. This partnership has been so successful that it is thought to have helped plants first colonize land hundreds of millions of years ago. Similarly, nitrogen-affixing bacteria form nodules on the roots of legumes, such as peas and beans, converting nitrogen from the air into forms the plant can use. Without this service, many plants would struggle to grow in nitrogen-poor soils. In the oceans, coral reefs are built on a symbiotic relationship between tiny coral animals and microscopic algae known as zooxanthellae. The algae live within the coral's tissues, capturing sunlight and producing sugars, much of which they share with their animal hosts. In return, the coral provides shelter and access to light, as well as nutrients from its own waste products. This exchange fuels the productivity of coral reefs, allowing them to thrive even in nutrient-poor tropical waters. The vibrant colors of healthy corals come from the algae themselves, and when environmental stress causes corals to lose these partners, the reef loses not only its beauty but much of its vitality. Symbiosis can take many forms. In mutualism, both partners benefit from the association, as in the cases above. In commensalism, one partner benefits, while the other is neither helped nor harmed. Some bacteria on human skin, for example, feed on oils and dead cells without noticeably affecting us. Parasitism, by contrast, benefits one partner at the expense of the other. While many parasites are larger organisms, some are microorganisms, including certain protozoa, fungi, and bacteria that live in or on hosts, drawing resources without giving anything back. Yet even parasitic relationships play a role in ecosystems, influencing the population sizes, behaviors, and evolutionary paths of their hosts. The boundaries between these categories are not always sharp. A relationship that is beneficial under one set of conditions may become harmful under another. For instance, microbes that normally live harmlessly in the gut can cause illness if they escape to other parts of the body or if the balance of the microbial community is disturbed. This fluidity reminds us that symbiosis is a dynamic interaction shaped by context, environment, and the changing needs of each partner. Microbial symbiosis extends beyond individual hosts to shape entire ecosystems. In soils, networks of bacteria and fungi connect the roots of different plants, allowing them to share nutrients and chemical signals. In deep-sea hydrothermal vents, giant tube worms survive without mouths or digestive systems because they house sulfur-oxidizing bacteria within their tissues. The bacteria convert chemicals from the vent water into organic matter, which feeds the worm. This is a form of primary production, entirely independent of sunlight, built on the metabolic power of microorganisms. Even within the human body, the microbiome behaves like a miniature ecosystem, complete with competition, cooperation, and checks on population growth. Different microbes produce substances that inhibit competitors or promote the growth of allies. Some species form biofilms, structured communities attached to surfaces that protect them from environmental stresses. The balance of the microbiome can be influenced by diet, lifestyle, antibiotics, and other factors, and when that balance shifts too far, it can affect the health of the host. Research continues to reveal just how deeply the microbiome is connected to processes as varied as digestion, immunity, and even mood regulation. Symbiotic relationships involving microorganisms have also played a role in major evolutionary transitions. The most profound example is the origin of eukaryotic cells themselves. According to the endosymbiotic theory, mitochondria and chloroplasts, the energy-producing organelles of eukaryotes, began as free-living bacteria that entered into a long-term partnership with another cell. Over time, these partnerships became so integrated that the symbionts could no longer live independently, and they became permanent residents within their host cells. This ancient symbiosis gave rise to the complexity of modern eukaryotic life, a reminder that cooperation between different organisms can lead to entirely new forms of existence. Symbiosis also teaches us about resilience and adaptation. Partnerships can allow organisms to survive in places where they could not live alone. They can open new ecological niches and create dependencies that bind species together in intricate webs. Yet these relationships can also be vulnerable. When environmental change disrupts a key partner, as rising sea temperatures do with corals and their algae, the entire system can unravel. Understanding these connections is essential not only for appreciating the beauty of nature's interdependence, but also for protecting it. The microbiome, whether in a human, a plant root, or a coral reef, is a living community whose members are in constant dialogue with each other and with their host. It is both delicate and robust, able to maintain balance under changing conditions yet sensitive to disturbance. To study these communities is to glimpse the deep truth that life is rarely solitary. Instead, it thrives through relationships, exchanges, and cooperation, whether at the scale of a forest or within the confines of a single cell. As we continue our journey through microbiology, we will move from these intimate partnerships to the broader stage of the environment itself. We will explore how microbes shape ecosystems, from the recycling of nutrients to the formation of soils and the regulation of Earth's atmosphere. In doing so, we will see how the influence of microorganisms extends far beyond individual hosts, touching every corner of the natural world. 10. Microbes in the environment. Everywhere life exists on Earth, from the deepest ocean trenches to the highest mountain peaks, microbes are there, quietly shaping the environment. They are not merely inhabitants of the planet, they are its unseen caretakers, constantly recycling materials, regulating chemical balances, and forming the foundation of ecosystems. Without them, the world would not resemble the hospitable place we know. Their work is so continuous, so pervasive, that it often passes unnoticed, even though it sustains the very conditions that make complex life possible. In soils, microbes are the unseen gardeners, breaking down fallen leaves, dead plants, and other organic matter into simpler compounds. This process of decomposition returns essential elements such as carbon, nitrogen, and phosphorus to forms that plants can use again. Bacteria and fungi work in concert with earthworms and insects, creating a network of nutrient cycling that enriches the soil and supports plant growth. Some microbes form intimate associations with plant roots, as nitrogen-affixing bacteria do in legumes, or as mycorrhizal fungi do in most plant species. These partnerships help plants extract nutrients more efficiently, allowing entire ecosystems to flourish. Without such microbial assistance, many landscapes would quickly lose their fertility. In aquatic environments, microbes form the base of the food web. Microscopic algae and cyanobacteria drift in the sunlit layers of oceans, lakes, and rivers, capturing light energy through photosynthesis and transforming it into organic matter. This material becomes food for zooplankton, small crustaceans, and other tiny grazers, which in turn feed larger animals. Through this chain, the energy captured by photosynthetic microbes flows upward to sustain fish, marine mammals, and seabirds. But the influence of microbes in water does not end with food production. In the depths, far below where sunlight reaches, Chemoautotrophic bacteria harness chemical energy from compounds like hydrogen sulfide, supporting entire communities around hydrothermal vents and cold seeps. These dark ecosystems, independent of the sun, are living examples of the versatility and resilience of microbial life. Microbes also play a central role in regulating the composition of Earth's atmosphere. Cyanobacteria were among the first organisms to produce oxygen through photosynthesis, fundamentally changing the planet's chemistry more than 2 billion years ago. Today, photosynthetic microbes in the oceans continue to contribute a substantial portion of the oxygen we breathe. Meanwhile, other microbes help control greenhouse gases. Certain soil bacteria consume methane, a potent greenhouse gas, reducing its release into the atmosphere. Nitrogen-cycling microbes convert nitrogen between different chemical forms, influencing plant growth, soil fertility, and the production of nitrous oxide, another greenhouse gas. Through these processes, microbes help moderate the planet's climate, acting as silent regulators of environmental stability. In the polar regions, microbes are active even in the coldest conditions. Beneath the ice, in briny channels or within frozen soils, they continue their slow cycles of metabolism, breaking down organic matter and influencing nutrient availability. In desert environments, microbial communities live in thin layers beneath the surface of rocks or within crusts on the soil, where they help stabilize the ground against erosion and capture moisture from rare rains. In every extreme, from boiling hot springs to acidic volcanic pools, microbes adapt their metabolisms to make use of whatever energy sources are available, shaping the local chemistry in the process. The impact of microbes extends into the geological realm as well. Some bacteria and archaea contribute to the formation of minerals, precipitating metals from solution, or altering the composition of rocks through their metabolic activity. Stromatolites, layered structures formed by microbial mats, are among the oldest known fossils, preserving a record of microbial life stretching back billions of years. Even today, similar microbial communities build stromatolite-like structures in certain shallow marine environments, continuing a process that has endured since the dawn of life. In addition to shaping the physical environment, microbes influence the health and resilience of ecosystems. They help decompose pollutants, breaking down oil spills, pesticides, and other contaminants into less harmful forms. This ability has been harnessed in bioremediation projects, where specific microbial species are encouraged to clean up contaminated soils or waters. Wetland microbes filter and purify water, removing excess nutrients and preventing harmful algal blooms. In these ways, microbes act as natural custodians, maintaining the balance of the environments they inhabit. Microbial communities are dynamic, shifting in response to environmental changes. Seasonal cycles, climate fluctuations, and human activity can alter the composition and activity of microbial populations. In turn, these shifts can have ripple effects throughout the ecosystem. For example, warming ocean temperatures can change the abundance and distribution of certain plankton species, affecting food availability for higher organisms. Changes in soil moisture can influence the balance of decomposers, altering the rate at which carbon is released or stored in the ground. Through these feedback loops, microbes link environmental change to ecological response. Their adaptability allows them to persist through both gradual shifts and sudden disturbances. After a volcanic eruption, microbes are often among the first organisms to colonize the new landscape, breaking down rock and beginning the slow process of soil formation. After floods or droughts, microbial communities adjust to the altered conditions, helping to restore the balance of nutrient cycles. In this way, they act as the foundation for ecological recovery, laying the groundwork for plants and animals to return. The environmental role of microbes is not limited to natural systems. Agricultural lands, urban soils, wastewater treatment facilities all rely on microbial processes to function. In farmlands, Microbes help recycle nutrients from crop residues and animal manure, maintaining soil fertility. In compost piles, they transform organic waste into rich humus. In wastewater plants, microbial communities break down organic matter and remove harmful compounds, making the water safe to return to rivers or seas. These humanity-managed systems mirror the natural cycles that microbes have been running for billions of years. To look at the environment through the lens of microbiology is to see a living network of invisible workers. They are constantly building, breaking down, transforming, and renewing the world around them. The air, water, and soil are not static backdrops for life, but active systems shaped in large part by microbial activity. Even when unseen, their presence is felt in every breath we take, every meal we eat, every drop of water we drink. Understanding the role of microbes in the environment deepens our appreciation for their quiet labor. It reminds us that the planet's health is intertwined with the vitality of these smallest of organisms. Protecting ecosystems means protecting the conditions in which microbial life can thrive, for without them, the cycles of life would slow and falter. In the next part of our exploration, we will step back from the present moment and consider the long history of humanity's relationship with microbes, not only as unseen partners in the environment, but as contributors to human culture, agriculture, and discovery. We will see how these hidden allies have shaped our history in ways we are only beginning to understand. 11. Microbiology in human history. For as long as humans have walked the earth, microorganisms have been our constant companions. Long before we could see them, long before we even imagined their existence, they were shaping the course of human life. Some influenced our survival in subtle and unseen ways. Others left their mark through the food we ate, the drinks we brewed, and the soil in which we grew our crops. The story of humanity is, in many ways, entwined with the story of microbes, though for most of history, we did not realize the depth of this connection. Early human societies unknowingly harnessed the power of microorganisms in their daily lives. Thousands of years ago, people learned that crushed grains left in water would bubble and produce a fermented, mildly alcoholic drink. This was the work of yeast, a microscopic fungus, converting sugars into alcohol and carbon dioxide. Bread making, too, was a microbial craft, long before it was a science. Leavened bread depends on yeast cells releasing gas as they digest sugars in flour, causing dough to rise into a light and airy texture. Across different cultures, fermentation became a way of transforming raw ingredients into foods with new flavors, textures, and longer storage life. Cheese, yogurt, kimchi, sauerkraut, soy sauce, and vinegar all owe their origins to the patient work of bacteria and fungi. These practices were developed through observation and tradition, passed down through generations, without a full understanding of the forces behind them. Ancient brewers, bakers, and farmers did not know the word microbe, yet they cultivated them through careful preparation and handling. The invisible world was at work in every jar of fermenting vegetables, every vat of brewing beer, every loaf of bread left to rise in the sun. In many cultures, these processes were seen as mysterious transformations, sometimes attributed to divine influence or the hidden power of nature. Microorganisms also played a role in agriculture from its beginnings. Even without knowing the science, early farmers benefited from the presence of nitrogeno-fixing bacteria in the soil, which enriched their fields and improved harvests. Rotating crops with legumes like beans and peas replenished soil fertility, a practice rooted in observation long before the discovery of the bacteria living in legume root nodules. Composting, the controlled decomposition of organic matter, was another ancient practice that relied on microbial activity to recycle nutrients back into the earth. While many of these relationships with microbes were beneficial, humans have also known the disruptive side of microbial life. Long before the germ theory of disease, people observed that illness could spread from one person to another, or follow certain conditions in food and water. Ancient civilizations developed practices of hygiene, food preservation, and quarantine, guided by patterns they could see even if the causes remained hidden. Boiling water, drying meat, salting fish, and fermenting milk were all ways of using environmental conditions to limit harmful microbial growth. The scientific understanding of microorganisms began to emerge slowly. The invention of the microscope in the 17th century by pioneers like Robert Hooke and Antony van Leeuwenhoek opened a window onto the hidden world. Leeuwenhoek, peering through his handcrafted lenses, described animalcules swimming in drops of water, scraping from teeth, and samples of food. Though he could not yet explain their role, his observations were the first clear evidence of a vast, unseen life surrounding us. Over the centuries that followed, scientists began to connect microorganisms with fermentation, decay, and eventually disease. In the 19th century, the work of Louis Pasteur demonstrated that fermentation was driven by living microorganisms, not by spontaneous chemical change. Pasteur also showed that heating food and drink could kill unwanted microbes, giving rise to the process we now call pasteurization. Around the same time, Robert Koch developed methods for isolating and identifying specific microbes responsible for particular diseases. His work provided the foundation for medical microbiology and confirmed that some illnesses were caused by specific microbial agents rather than by vague environmental factors. These discoveries transformed medicine, agriculture, and industry. Understanding the role of microbes in disease led to improvements in sanitation, hygiene, and water treatment. The development of vaccines, many of which use harmless or weakened microbes to train the immune system, became a powerful tool for preventing illness. The discovery of antibiotics in the 20th century, beginning with Alexander Fleming's penicillin, revolutionized the treatment of bacterial infections. In agriculture, knowledge of soil microbiology informed better farming practices, crop management, and the development of microbial inoculants to enhance plant growth. Beyond health and farming, microbes became indispensable in industry. Fermentation processes were refined for large-scale production of food, beverages, and pharmaceuticals. Microbes were used to produce enzymes for detergents, biofuels from plant matter, and biodegradable plastics. In biotechnology, genetically engineered bacteria began producing insulin for people with diabetes, along with a wide range of other medicines and industrial products. These applications highlighted the versatility of microorganisms and their ability to serve as tiny factories for human needs. In more recent decades, the study of the human microbiome has reshaped our view of health and disease. We now know that the communities of microbes living within us are not passive passengers, but active participants in digestion, immunity, and even mood regulation. This understanding has inspired new approaches to medicine, such as probiotics and microbiome-focused therapies, which seek to maintain or restore healthy microbial balance. The recognition that our bodies are ecosystems in their own right has brought us full circle to an ancient truth, that life thrives in connection, not isolation. Yet our relationship with microbes is not one of control, but of mutual influence. Human activity can change microbial communities in ways that echo back to us. The overuse of antibiotics, for example, can disrupt beneficial microbes and encourage resistant strains. Agricultural practices can alter the microbial life of soils, affecting fertility and resilience. Climate change influences microbial activity in oceans, wetlands, and permafrost, with consequences for carbon and nutrient cycles. Our shared history with microbes continues to evolve, shaped by both our actions and their adaptations. Looking back, it is humbling to realize that microorganisms have been part of every stage of human development, from the earliest loaves of bread baked in clay ovens to the latest genetic engineering in high-tech laboratories. They have nourished us, healed us, challenged us, and changed us. They are our oldest collaborators and our most enduring companions. The history of humanity is not separate from theirs. It is a shared story of coexistence, adaptation, and transformation. As we move forward in our journey through microbiology, we will now turn to the tools that have allowed us to study this hidden world in such depth. We will explore how the development of microscopes and other technologies has expanded our ability to see, measure, and understand microbes, opening doors to discoveries that continue to shape science and society. 12. Microscopes and modern microbiology. The journey into the world of microbiology is, at its heart, a journey of vision, of learning to see what was once invisible. Our understanding of microorganisms has grown in step with the tools we use to observe them, and chief among these tools is the microscope. It is the lens, both literal and metaphorical, through which we have come to recognize the richness and complexity of life at its smallest scales. Without it, microbes would remain hidden, and our knowledge of biology, medicine, and the environment would be incomplete. The earliest microscopes were simple devices, born from the same curiosity that led artisans to fashion lenses for spectacles in the late Middle Ages. These early instruments could magnify objects only modestly, but they hinted at the possibility of exploring a smaller, unseen world. In the 17th century, this possibility became a reality through the work of pioneers like Robert Hooke and Antony van Leeuwenhoek. Hooke, in his groundbreaking work, Micrographia, used a compound microscope to study the structure of plants, insects, and minerals. His drawing of cork, showing its honeycomb of cells, captured the public imagination and introduced a term that remains central to biology today. Leeuwenhoek's approach was different, but equally transformative. Working alone in the Netherlands, he crafted single-lens microscopes of remarkable clarity, capable of magnifications far greater than most instruments of his time. Through these simple but powerful devices, he observed an astonishing variety of microscopic life bacteria, protozoa, sperm cells, and more. He called these organisms animalcules, little animals, and described their movements and behaviors with care. Leeuwenhoek's meticulous observations revealed, for the first time, that the world was teeming with life, far too small to see unaided. In the centuries that followed, microscope design advanced steadily. Compound microscopes, using multiple lenses to achieve higher magnification, became standard in scientific study. Improvements in lens grinding and mounting reduced distortion and increased clarity. By the 19th century, microscopes could reveal the internal structures of cells, allowing scientists like Matthias Schleiden and Theodor Schwann to formulate the cell theory, the principle that all living things are composed of cells and that cells are the fundamental units of life. These insights laid the foundation for modern biology and medicine. Yet even the best light microscopes have limits. Because they rely on visible light, they cannot clearly resolve objects smaller than about 200 nanometers. Many structures within cells and the smallest viruses are far smaller than this. To go beyond these limits, scientists needed a different approach. This came in the 20th century with the invention of the electron microscope. Instead of light, electron microscopes use beams of electrons, which have much shorter wavelengths, allowing resolution of structures only a few nanometers across. The electron microscope opened new realms of discovery. Transmission electron microscopes, TEM, could reveal the fine details of cell interiors, the membranes, ribosomes, mitochondria, and intricate folds of internal structures. Scanning electron microscopes, SEM, produced detailed three-dimensional images of cell surfaces, showing the textures and contours of microscopic forms in stunning clarity. These images transformed our perception of microbes, showing them not as vague specks but as complex, beautifully structured entities. As technology advanced, new forms of microscopy emerged, Fluorescence microscopy introduced the use of dyes and fluorescent proteins that could bind to specific molecules within cells, making them glow under certain wavelengths of light. This allowed scientists to track the location and movement of proteins, trace the paths of individual molecules, and visualize interactions in living cells in real time. Confocal microscopy refined this approach by using lasers to produce sharp, three-dimensional images, eliminating much of the blur caused by out-of-focus light. In recent decades, super-resolution microscopy has pushed the boundaries even further. Techniques such as STED, stimulated emission depletion, and PALM, photoactivated localization microscopy, have broken the diffraction limit of light, achieving resolution down to tens of nanometers. These advances have brought into view structures and processes that were once beyond our reach, deepening our understanding of how microbes function at the molecular level. Microscopy has also grown more accessible. Once limited to specialized laboratories, high-quality microscopes are now found in schools, field research stations, and even in portable forms that can be used in remote locations. Digital imaging allows scientists to share their observations instantly across the globe, enabling collaboration and accelerating discovery. The integration of microscopy with computational analysis has opened new possibilities for studying vast datasets, tracking microbial populations, and modeling their behaviors. Modern microbiology depends not only on the ability to see microbes, but also on the ability to connect what we see with what we know about their genetics, chemistry, and ecology. Microscopy is often combined with molecular techniques such as DNA sequencing, which can identify species and reveal their evolutionary relationships. Imaging can show where a microbe lives and how it interacts with its neighbors, while genetic analysis can explain the capabilities and adaptations that allow it to thrive in that environment. Together, these tools create a fuller picture of microbial life than either could provide alone. Beyond the laboratory, microscopy has found important applications in medicine, agriculture, and environmental science. In clinical settings, it remains a primary method for diagnosing infections, allowing doctors to see bacteria, fungi, and parasites directly in patient samples. In agriculture, microscopes help identify plant pathogens, guiding efforts to protect crops. In environmental monitoring, microscopy reveals the presence and abundance of plankton, tracking changes in ecosystems that can signal broader environmental shifts. The microscope is more than an instrument. It is a way of thinking, a means of engaging with the hidden structures of the natural world. Each advance in its design has opened a new chapter in our understanding of life's diversity and complexity. It reminds us that the boundaries of our knowledge are often set by the limits of our perception, and that expanding those limits can transform our understanding of the world. Today, as we look into the eyepiece or at a computer screen, displaying the magnified image of a microbe, we are participating in a long tradition that began with the curiosity of early lens makers. We are peering into a world that is at once ancient and ever new, where each observation offers the chance to uncover something never before seen. The microscope has given us the ability to witness the beauty and intricacy of the smallest forms of life, and in doing so, it has revealed the profound truth that complexity exists at every scale. In the next part of our journey, we will step beyond Earth's familiar environments and into the realm of speculation, exploring the possibilities of microbial life beyond our planet. We will consider what we have learned about the resilience of microbes here and how that shapes our search for life among the stars. 13. Microbes in space and the search for life. When we lift our gaze from the Earth to the vastness of the night sky, we are reminded that our planet is but one small world among countless others. The question of whether life exists beyond Earth has captured human imagination for centuries, and in the modern era, it has become a subject of scientific inquiry. In searching for life elsewhere, we begin with the smallest and most adaptable forms we know, the microorganisms. If life exists beyond our planet, it is likely that it began, and perhaps still exists, in microbial form. Their resilience on Earth suggests they could survive in environments far more extreme than those that support larger, more complex organisms. On our own planet, microbes thrive in places that seem hostile to life. They inhabit boiling hot springs, acidic pools, frozen tundra, and the deep subsurface where sunlight never reaches. Some live in hypersaline lakes with salt concentrations higher than seawater, others in environments saturated with toxic metals, and still others in rocks kilometers beneath the surface. Certain bacteria can endure intense radiation, surviving doses hundreds of times greater than what would be lethal to humans. Others form durable spores that can lie dormant for centuries, awaiting more favorable conditions. This adaptability suggests that if similar environments exist elsewhere in the solar system or beyond, microbes could potentially survive there. Mars has long been a focus of the search for extraterrestrial life. Today it is cold and dry, with a thin atmosphere and surface bathed in radiation. Yet evidence from spacecraft and rovers points to a different past. Billions of years ago, Mars appears to have had liquid water on its surface, rivers, lakes, and perhaps even shallow seas. If life ever arose there, microbial forms would have been the most likely inhabitants. Scientists have found signs of ancient riverbeds, minerals that form in water, and chemical signatures that might hint at organic molecules. While no conclusive evidence of life has yet been found, the possibility remains that Mars could harbor dormant microbes underground, protected from radiation and occasional temperature extremes. Beyond Mars, the icy moons of the outer solar system offer intriguing possibilities. Jupiter's moon, Europa, and Saturn's moon, Enceladus, both have thick ice shells covering global oceans of liquid water. Beneath their frozen surfaces, heat from tidal forces keeps these oceans from freezing solid. Plumes of water vapor and ice particles have been observed erupting from Enceladus, carrying organic molecules from the subsurface ocean into space. These oceans, shielded from harsh radiation by ice and enriched by minerals from the rocky interiors below, could provide environments where microbial life might thrive. On Earth, deep-sea hydrothermal vents host rich microbial communities without any reliance on sunlight, drawing their energy from chemical reactions between seawater and hot rock. Similar processes could be occurring in the hidden oceans of these distant moons. Another candidate is Titan, Saturn's largest moon, whose thick atmosphere and lakes of liquid methane and ethane present a very different chemistry from Earth. While the extreme cold and lack of liquid water make Titan an unlikely match for life as we know it, some scientists speculate that exotic microbes could exist there, using methane in place of water and hydrocarbons in place of organic molecules familiar to us. If such life exists, it would expand our understanding of what is possible for living systems. The search for microbial life beyond Earth is not limited to our solar system. With the discovery of thousands of exoplanets, planets orbiting other stars, astronomers have identified worlds that may have conditions suitable for life. Some lie within their star's habitable zone, where temperatures could allow liquid water to exist. While we cannot yet see these planets in detail, future telescopes may be able to detect atmospheric gases that could hint at biological activity, such as oxygen, methane, or combinations of gases unlikely to persist without a continuous source. Microbial life, if present, could subtly shape the chemistry of these distant worlds in ways detectable from afar. Microbes have also become central to the concept of panspermia, the idea that life can spread between planets, or even between star systems. Meteorites from Mars have been found on Earth, blasted into space by ancient impacts, and eventually falling through our atmosphere. Experiments have shown that some microbial spores can survive the vacuum of space, intense radiation, and extreme temperature changes for extended periods, especially if shielded by rock or dust. This raises the possibility that life could travel between worlds, carried on debris from planetary collisions. While panspermia does not explain the origin of life itself, it suggests a mechanism by which life could spread once it begins. In addition to searching for life elsewhere, scientists study how microbes respond to space itself. On the International Space Station, experiments have shown that some microbes can grow and reproduce in microgravity, and certain bacterial species even become more resistant to stress. Others form biofilms more readily in space, which has implications for both spacecraft maintenance and the survival of microbes on long journeys. Understanding these adaptations helps prepare for future missions and informs the question of whether microbes could endure long interplanetary voyages. The possibility of extraterrestrial microbes also shapes how we explore other worlds. Space agencies follow strict planetary protection protocols to avoid contaminating potentially habitable environments with Earth's microbes, which could confound the search for native life. Likewise, precautions are taken to prevent bringing back unknown organisms that might be hazardous. Samples returned from the Moon, Mars, or other bodies are handled with great care, studied in secure laboratories, to ensure both safety and scientific integrity. The search for microbial life beyond Earth is as much about perspective as it is about discovery. Even if we never find life elsewhere, the effort deepens our appreciation for the resilience of life here. It reminds us that microbes are not just inhabitants of Earth, but examples of a universal potential, the ability of life to adapt, endure, and perhaps arise wherever conditions permit. If life is found beyond our planet, it will likely begin with the discovery of something small, a cell, a fossilized microbe, a chemical signature pointing to metabolic activity. Yet such a discovery would be profound, showing that life is not unique to Earth, that the spark of biology can ignite in more than one place in the cosmos. Whether in the salty brines of Mars, the hidden oceans of icy moons, or the distant planets orbiting other stars, the search continues. And as we learn more about the microbes of Earth, their adaptability, their persistence, their ingenuity in finding ways to live, we refine our picture of what might be possible elsewhere. Microbes may hold the key not only to our understanding of life's beginnings, but also to its distribution in the universe. In the next part of our exploration, we will turn our attention back to Earth and reflect on the unity of life at the microscopic scale, tracing the connections between all living things and the shared heritage that links the smallest bacterium to the largest tree or whale. This unity, revealed through the study of microbiology, is a reminder of the deep kinship that underlies the diversity of life. 14. The unity of life at the microscopic scale. When we look closely at the world of microbes, and indeed at all living things, we begin to see a profound truth. Life, in all its variety, is built from the same fundamental patterns. From the simplest bacterium drifting in a drop of water to the largest whale gliding through the ocean, every organism shares a common heritage. This unity is not immediately obvious when we consider the differences in form, size, and complexity, but at the microscopic level, the similarities shine through. All life is made of cells. All cells carry genetic material, and all use similar chemical pathways to sustain themselves. The idea that all living things are related, that they descend from a shared ancestor, is one of the most powerful concepts in biology. It means that the vast diversity we see today, the forests, the coral reefs, the grasslands, and the teeming invisible world of microbes, are all branches of the same great tree of life. This tree reaches deep into the past, billions of years ago, to the first cells that managed to capture energy, store information, and replicate themselves. Though we cannot see those earliest forms, we see their legacy in the universal features of life today. One of the strongest threads connecting all organisms is the genetic code. In every known form of life, Information is stored in long molecules of nucleic acids, DNA, or in some viruses, RNA. This information is written in a four-letter alphabet of chemical bases, and the meaning of these letters is the same for all species. The sequence of bases forms genes, each carrying the instructions to build a specific protein. Proteins, in turn, are the workhorses of the cell, shaping its structure, driving its reactions, and regulating its functions. The universality of the genetic code is a sign of common descent. It is the shared language of life, spoken by bacteria, oak trees, and humans alike. Beyond the genetic code, there is the shared machinery that interprets it. All cells use ribosomes, complex molecular machines, to read the instructions in RNA and assemble amino acids into proteins. The process of transcription, in which DNA is copied into RNA, and translation, in which RNA is used to build proteins, is remarkably similar across all forms of life. The biochemical pathways that generate energy, such as glycolysis, are also conserved, evidence of their origin in a distant common ancestor. Even the membranes that enclose cells are built from related kinds of lipid molecules, forming barriers that protect the delicate chemistry of life. This unity extends to the logic of how cells operate. All living things must obtain energy, build the molecules they need, sense their environment, and respond to changes. Microbes do this with astonishing efficiency, but the same principles are at work in the cells of a hummingbird or a human being. When we study how a bacterium regulates its genes or repairs its DNA, we are often learning about processes that occur in our own cells as well. In this way, microbiology is not just the study of the small, it is the study of the shared foundation of all life. The unity of life is also reflected in its adaptability. While the forms and strategies of different species vary, the capacity for change is universal. Life evolves by variation and selection, shaping organisms to fit their environments. The rapid reproduction of microbes allows us to watch evolution in action, but the same process, stretched across longer timescales, has shaped every species on Earth. The beak of a bird, the leaf of a plant, the fin of a fish, all are the products of this shared evolutionary mechanism, working with the same basic building blocks. Recognizing this unity changes the way we see the natural world. It blurs the boundaries we often place between us and them, between the visible and invisible, between the simple and the complex. We come to understand that the difference between a microbe and a human is not a difference of kind, but of degree. The same principles that allow a bacterium to swim toward food or away from harm also guide the cells in our own bodies as they respond to signals and maintain balance. The kinship is deep, even if it is hidden from casual view. This perspective also brings humility. Humans often think of ourselves as standing apart from the rest of life, but microbiology reminds us that we are part of a continuum. We carry within ourselves the molecular echoes of ancient microbes. The mitochondria that generate our energy were once free-living bacteria, now so integrated into our being that we could not live without them. The chloroplasts in plants, which capture sunlight and make food, share a similar history. Our own DNA contains sequences inherited from ancient viruses that once infected our ancestors. We are, in a very real sense, mosaics of life's history. The unity of life also offers hope for discovery. Because the same fundamental processes occur in all organisms, what we learn from one can often be applied to others. The study of yeast has illuminated the workings of human genes. Research on bacterial enzymes has led to breakthroughs in biotechnology. Insights from the immune systems of plants and microbes have inspired new medical therapies. This transfer of knowledge is possible because of the shared heritage that binds all life together. In the grand sweep of time, this unity is a reminder that life's story is a shared one. Every organism alive today is connected to every other by an unbroken chain of descent. This connection is not just a fact of history, it is an active reality. The oxygen you breathe was made by photosynthetic microbes. The nutrients in your food were cycled by bacteria and fungi. The very cells that make up your body operate according to rules refined over billions of years in the laboratories of the natural world. To see life in this way is to see a kind of quiet harmony underlying its diversity. It is to recognize that the swirling complexity of ecosystems, the vast differences in form and function, are all expressions of a single ancient theme. The smallest bacterium and the tallest tree are variations on a shared design, each carrying forward the legacy of that first spark of life. As we near the end of our journey through the fundamentals of microbiology, this unity stands as one of its most profound lessons. The microbial world is not separate from the rest of life. It is its foundation. By studying the smallest forms, we come to understand the principles that govern all living things, including ourselves. We see that the boundaries between species are bridged by shared chemistry, shared information, and shared history. In the final part of this exploration, we will step back and reflect on what we have learned, considering the quiet persistence of microbial life across the ages and the deep sense of connection it offers to the whole of the living world. This gentle closing will bring our journey to rest, leaving us with the awareness that even in stillness, life continues its subtle and eternal work. 15. Closing thoughts and gentle farewell. As we reach the end of our quiet journey through the fundamentals of microbiology, it is worth pausing to take in the vast landscape we have traveled. We began by stepping gently into the hidden world of microbes, a realm too small for the naked eye yet immense in its variety and significance. Along the way, we met the ancient prokaryotes, the adaptable bacteria and archaea that laid the foundations for life as we know it. We explored the more complex eukaryotic microbes, whose internal structures opened the door to new strategies for living. We lingered at the edge of life with viruses, those mysterious carriers of genetic information that blur the boundary between living and non-living. We followed the pathways of microbial metabolism, the quiet currents of energy that sustain all cellular life, and watched populations grow, divide, and adapt through reproduction. From there, our view widened to see how microbes live not in isolation, but in connection, forming microbiomes, engaging in symbiosis, shaping ecosystems in ways both visible and invisible. We looked at their environmental roles, recycling nutrients, balancing the atmosphere, and supporting every living thing on the planet. We traced humanity's relationship with microbes through history, from ancient bread and fermented drinks to the sophisticated science of modern microbiology. We looked through the lens of the microscope, seeing how each advance in technology deepened our understanding. We ventured outward into space, considering the possibility that microbes may dwell beyond Earth, and we reflected on the unity of life, the shared chemistry and common heritage that bind together every living organism. Through it all, a pattern has emerged, one as gentle and persistent as the flow of a quiet river. Microbes are not simply background actors in the drama of life. They are the stage itself, the conditions that make life possible. They are the recyclers, the builders, the invisible threads weaving together the fabric of ecosystems. They remind us that strength is not always a matter of size, and that complexity can grow from the smallest beginnings. It is easy to overlook this world. We cannot see it without help. We do not feel its constant work. Yet our very existence depends on it. The air we breathe, the food we eat, the health of our bodies, and the balance of our planet, all are shaped by the quiet activity of microbial life. Even now, as you listen in stillness, countless microbial processes are unfolding around you and within you. They are turning sunlight into energy in the leaves outside. They are cycling carbon in the soil. They are digesting the food in your stomach, producing vitamins, training your immune system. The microbial world is always at work, steady and unhurried. In learning about microbes, we are reminded of the deep connections that link us to the rest of life. We are not separate from this invisible world, but part of it. Our own cells carry echoes of ancient microbial partnerships, mitochondria, and chloroplasts that began as independent bacteria, now inseparably woven into the fabric of our being. The DNA in our chromosomes shares a common language with the simplest bacterium. In this way, every breath, every heartbeat, every step we take is part of a continuum stretching back to the first cells that ever lived. There is also a kind of comfort in this perspective. Microbes have endured for billions of years, surviving mass extinctions, dramatic shifts in climate, and the slow rearrangement of continents. They have adapted to every challenge, finding ways to persist in conditions that seem unlivable. Their resilience is a quiet assurance that life, in some form, will continue, whatever changes the Earth may face. And just as they have shaped the planet in the past, they will continue to do so long after our own species has played its part. As our exploration draws to a close, we might imagine the microbial world not as something distant or abstract, but as a living presence all around us in the soil beneath our feet, in the water that flows through rivers and seas, in the air that drifts across the land, and even in the spaces between our cells. It is a world that hums softly with the rhythm of life, unseen but constant, intricate yet harmonious. In its quiet way, it is a reminder that the most profound forces are often the ones we do not notice, the ones that work without fanfare, patiently sustaining the world. To carry this awareness with us is to see life differently. It is to look at a blade of grass and know that its green comes from the work of microscopic chloroplasts. It is to see a loaf of bread and remember the yeast cells that made it rise. It is to walk through a forest and understand that every leaf that falls will be broken down, piece by piece, by countless unseen decomposers, their work returning nutrients to the soil. It is to see ourselves not as solitary beings but as part of a vast and interwoven network of life. In this gentle understanding lies a kind of peace. We are surrounded by life's persistence, by the patient work of countless small beings who ask nothing from us but the space to live. They remind us that we are sustained not only by the visible gifts of the world, but by its invisible ones as well. In the grand and ancient story of life on Earth, we are newcomers, guests in a home long tended by its smallest inhabitants. So as we bring our journey to rest, we do so with gratitude for the microbes that breathe with the trees, that stir in the depths of oceans, that move through the soil, that live within us. They have been here from the beginning, and in their quiet way, they will be here until the end. The study of microbiology opens a window onto this hidden world, but the world itself has always been here, always working, always waiting to be noticed. In the stillness now, as you drift toward rest, you might imagine that vast, unseen world continuing its patient rhythms, the rivers of energy flowing from sunlight to sugar, from sugar to motion, the endless cycles of growth and decay, of creation and renewal, the deep, unbroken thread that ties all living things together. It is a world that asks nothing more than to be understood, and in understanding, to be appreciated. Our exploration ends here, but the world of microbes goes on, unending, unfading, forever part of the quiet heartbeat of the earth. And in knowing this, perhaps we can rest a little easier, feeling ourselves part of something vast, enduring, and profoundly connected.